Hi, everyone. Welcome to this Thermo Fisher webinar series. My name is Dr. Ernesto Goulart, and I'm thrilled to be here today to present to you a little bit of the work that I've been doing in Brazil over the last 5 years on stem cell research, on exciting in stem cell research. So first of all, I'd like to thank the invite from Thermo Fisher, especially Chelsea, Jennifer, and Marlene from Thermo Fisher Brazil. It's a pleasure to be here. And so today's talk, I'm going to talk to you guys a little bit of how to use iPS-derived cells to generate a fully functional human liver using 3D bioprinting. So all of the work that I've been going to show you today is published work that I developed during my PhD at the University of São Paulo at the Human Genome and Stem Cell Research Center. So, first of all, this is my disclosure that I want to make sure to pop out. It's that I declare that I'm not receiving any financial or material retribution to participate in this webinar. So the agenda of this presentation is divided basically on 3 sections. So first, I'm going to introduce you how and why I chose to work with liver tissue engineering, a little bit of iPS cell culture and iPS cell quality control as well, and go to the functional phenotype differentiation protocols. So this is very important because I think the keynote that I want to show you guys is how to properly phenotype your differentiated stem cells. So I'm going to show you how to functionally phenotype hepatocytes using 2D differentiation, also endothelial cells and mesenchymal cells. And finally, I'm going to show you how we derive this differentiation protocol for 3D hepatic differentiation. And finally, I'm going to show you a little bit of the results and future perspective on the liver tissue engineering field. So the liver is the larger internal organ of the body. So it has a lot of functions. So we all know that the liver is responsible for the metabolism of carbohydrates, lipids, nitrogen compounds, but also performs important function in blood homeostasis, such as hemostasis and the control of hemodynamics. But also liver performs a very important function, which is a biotransformation. So basically everything that we absorb in the intestines is transformed in the liver to avoid toxic effects of the molecules that we absorbed. So this is performed majorly by the C450, is the main enzyme of this complex that performs this biotransformation. But also the liver performs an important role in the cholesterol and lipoprotein metabolism, such as performs the fat absorption through the bile secretion in the intestine, but also in the LDL metabolism. Liver performs more than 500 different physiological functions. So basically the liver is a metabolic hub of our body. So in terms of regeneration, the liver is a very interesting organ. So the liver possesses this amazing ability to regenerate itself. And this is actually known back since the Greek mythology. As you can see here on this picture on the top left, there's the Prometheus myth in the Greek mythology that tells us a story of this titan called Prometheus that was doomed by the gods to be attached in the top of a mountain where every day an eagle will eat part of his liver, and the next day the liver will grow back, and the eagle will do that again over and over throughout the eternity. And this ability to regenerate itself is quite known for decades. So as you can see here on the bottom image, after a partial hepatectomy performed in mice, after 7 days, the liver goes back to its normal size and also its shape as well. So this is a very complex and timely regulated process of regeneration. But this is very, very interesting in terms of regenerative medicine. But although the liver possesses this amazing ability to regenerate, many diseases can evolve from chronic exposure to hepatotoxins. So chronic liver disease affects more than 60 million people only in US and Europe. And the liver cirrhosis, which is the chronic deposition of scar tissue within the liver, is associated with 2% of global death burden, which is approximately responsible for 1 million deaths a year. Another process that is resulted from chronic inflammation of the liver parenchyma is called liver steatosis. So steatosis is the accumulation of fat within the parenchyma. It was usually associated with alcohol consumption, but due to this global-wide pandemic of obesity and diabetes, non-alcoholic fatty liver disease is estimated to affect 20% of the global population. And the fat accumulation within the liver parenchyma could lead to what we call NASH, which is non-alcoholic steatohepatitis, which is the trigger of inflammation of the liver parenchyma due to this fat accumulation on the tissue. And these two processes, these two pathologies, the cirrhosis and steatosis, could lead to the development of cancers such as hepatocellular carcinoma, which is the fourth leading cause of death among all tumors. And just so you know, hepatitis C, which is one of the main pathogens that could lead to this excessive inflammation of the liver parenchyma, is estimated to infect more than 80 million people in the world, 1.6 million only in Brazil. So when the liver reaches that chronic stage, it cannot grow back and cannot regenerate itself. So what can we do to treat patients with that condition? So basically there's one available option, which is transplantation. It could be either a whole liver transplantation or a partial liver transplantation between two live individuals. So although there were many developments in the surgical field on the organ transplantation research field, many of the same challenge that we faced back in 1954, where the first transplant was performed, we still face it today, which is a donor shortage, immune compatibility issues, rejection, and also a lifelong immunosuppressor therapy. So it is estimated to be performed around 25,000 liver transplantations every year in the world. In Brazil, in 2017, we have more than 5,000 patients in waiting list. At that same year, we have almost 3,000 new entries and only performed 2,000. So this data shows us that there is a huge gap between the need and the availability of organs for transplantation. As you can see here on this data from the US, where we can see that the gap between organs for transplantation and patients that need is still increasing over the years. So we need to do something for address this issue, and that's something that I chose to work in my scientific career to develop new technologies to address this important issue. So what I'm going to talk to you today is how to use stem cells to generate functional tissue for transplantation. So basically, we're using this technology of cell culture that was almost 2 centuries old and applying new technologies and new exciting discoveries that was performed over the last few years that could generate a fully functional and complex tissue that could one day meet the need for liver transplantation. So basically, liver tissue engineering and tissue engineering in general are basically depending on 2 important factors. So we need a scaffold where the cells will grow onto, and also we need a proper cell source. So we need cells and scaffold to generate a fully functional tissue engineering graft. So let's talk first on cell source. So what are the cell source that we can use for liver tissue engineering strategies? So there are several options. So of course we have primary human hepatocytes, which are hepatocytes that are obtained from diseased liver. They are highly functional because they are obtained from an adult individual. However, since they are terminally differentiated cells, they do not proliferate very well in culture, and they will always generate heterologous transplantation. So you will still face the same issues of immune rejection and immune suppressor therapy. But we also have what we call transdifferentiation protocols, which is the introduction of some few genes that could transform perhaps a fibroblast towards an hepatic phenotype, such as hepatocytes. And this transdifferentiation protocol, it leads to the generation of very highly functional hepatocytes, but they still have very low proliferative ability. This protocol could generate autologous transplantation, but as I mentioned before, the liver is the larger internal organ of the body, so we need a lot of cells to generate a very important liver function to meet the needs of patients with chronic liver disease. But we do have also stem cells. So we have embryonic stem cells that once you differentiate the embryonic stem cells that have this plasticity that can generate any cells of your body, the cells at the end of the differentiation, they do have a moderate functionality. That means that the hepatocytes that are differentiated from embryonic stem cells, they're not fully mature. They are more like a fetal phenotype. But the embryonic stem cells are highly proliferative cells. So we can generate tons of cells within days. And that's very important for the liver, as I mentioned. However, embryonic stem cells will always generate heterologous transplantation. But we do have iPS cells. So iPS cells are pluripotent stem cells that are generated from a process called reprogramming, which is we take a primary cell and then transform it back to a pluripotent state, where at that time the cells can differentiate towards any phenotype that we want. So we have this plasticity of the embryonic stem cell. The differentiated cells from iPS do also have this moderated functionality as well. They are highly proliferative and they can generate autologous transplantation. What does that mean? So basically, I can make iPS cells from any individual that I want. So I can make iPS cells from me, from you, anyone. So this is very interesting because this can generate autologous transplantation. So what we did establish in our laboratory is a protocol that we can take blood from patient. It's a simple blood sample collection. And then we can isolate erythroblast from that patient. And after the process using the reprogramming factors, call it Yamanaka's factor, we can generate iPS cell lines from that same individual. And then we established three different differentiation protocols. So what are the cells that we need to generate a liver? So basically, there are three main cellular phenotypes that we need. We need hepatocytes, endothelial cells, and mesenchymal cells. So these are the protocols that we establish in our laboratory. So basically, hepatocytes go first towards definitive endoderm, then hepatoblast, and then finally hepatocytes within 18 days. Endothelial cells, on the other hand, the vascular cells, are derived from mesoderm, and then they are differentiated towards endothelial progenitor cells, where we sort the cells for CD31 positive cells, and then differentiate it finally towards endothelial cells. Well, we also need stromal cells. And for that, we establish a mesenchymal differentiation process where we go first towards neurocrest. So basically, the stromal cells that we derived here in our work is more craniofacial mesenchymal stem cells. So okay, we do have now all the cells that we need to generate a liver. So how can we apply that cells in a scaffold to generate a fully functional liver tissue? So in our laboratory, we work with basically two different technologies. We use 3D bioprinting, but we also work with organoids. So organoids are self-assembled tissues that recapitulates early stages of organogenesis. So basically, if you give the proper factors and the proper environment to these three cells that I mentioned in the previous slide, they start spontaneously to organize and form this tissue that we call liver organoids that closely resemble the liver bud during the early stages of the liver organogenesis. But today I'm going to talk about the 3D bioprinting strategy that we recently published. So if you're interested in getting to know a little bit of the organoid work that I've developed over the last few years, you can find on this publication on the top right. So let's talk about the 3D bioprinting. So 3D bioprinting usually uses a single cell dispersion within an hydrogel. So the 3D bioprinting is a process where there's a several layer deposition of a biomaterial, and this biomaterial is basically an hydrogel, so it's basically a gel that could be composed by extracellular matrix perhaps, and cells, so the cells that we differentiate. So once you put this multi-layer structure, it can give you a 3D shape that you want. So basically all the most used 3D bioprinting process use this a single cell dispersion. So you take the cells out of the culture and then put it in an hydrogel. However, importantly, the hepatocytes, which is the more important liver parenchymal cells, they are epithelial cells. And as we all know, epithelial cells like to stay in close contact to each other. If they don't do that, they could underwent what we call epithelial mesenchymal transition. So basically, the cells lose the epithelial phenotype and go towards a more mesenchymal phenotype, where the cells can move throughout the environment. And once they do that, usually they lose the phenotype, the original phenotype. So this is very critical for the liver perspective, because if you lose the hepatocytes phenotype, there is no liver function whatsoever. So to address this issue, we evaluate the use of what we call spheroids. So spheroids are a 3D organization of cells that closely keeping in close contact the epithelial cells. So basically all the hepatocytes are going to be close to each other, in close contact to each other, and they will eventually retain this epithelial phenotype. So instead of using single cell dispersion within hydrogel, can we use hepatic spheroids and disperse the spheroids within the hydrogel to make a more functional liver tissue? So that's something that we decided to investigate it and got very interesting results. So let's first talk about iPS cell lines quality control. So let's first check if the iPS cells that we derived are indeed iPS cells. So to do that, first we need to, of course, check for chromosomal abnormalities. So we perform what we call MLPA for subtelomeric regions, and then we evaluate, we show that there is no trisomy or any chromosomal abnormalities whatsoever. But we also check the expression of important pluripotent markers such as OCT4 and SAC4 on the bottom left here. And also for iPS functional analysis, we need to show that the cells that we generate have the ability to differentiate towards the three germlines, so endoderm, mesoderm, and ectoderm. You can do that by applying a teratoma assay as we do here, but you can also use an animal-free method that we can spontaneously differentiate the cells and check the expression of important germline markers. So as we can show here, the iPS that we derive are fully functional iPS cell lines with no chromosomal abnormalities. So next we go to functional phenotype, the differentiation process that we need to use in order to generate this liver tissue. So as I mentioned, we have hepatocytes, endothelial cells, and mesenchymal cells. So let's see if the hepatocytes that we differentiated are fully functional hepatocytes. So first, let's view the 2D hepatocyte differentiation. So as you can see here on the top left, we check the expression of important hepatic markers such as cytokeratin 18, glycol 6-phosphatase, and albumin and UGT1 throughout the differentiation process by RT-qPCR. And we see the increase of hepatic differentiation throughout the differentiation process. At the very end of the differentiation, we stain the cells for flow cytometry for albumin and UGT1, and we can see that almost 80% of our population express these two important hepatic markers. But we also need to see if the hepatocytes still have the functional ability to perform important hepatic functions. So on the bottom image, we can see an assay where we put this dye called ICG, and this green dye is transported into the hepatocytes and then transported back to the lateral plate of the hepatocytes, which is a process called basolateral transportation. So after 1 hour, the hepatocytes uptake this dye, and then after 4 hours, they release it back to the media, which shows that the hepatocytes have this ability to perform basolateral transportation. And also we check the expression by immune fluorescence of important hepatic markers such as CYP3A4, icadherin, alpha-1 antitrypsin, and albumin as well. And finally, we check the expression of important hepatic transaminases such as GSTA1, GSTA4 throughout the differentiation process. And also, we checked if there is the cells that we derive it have the ability of respond to important drug inducers such as omeprazole and rifampicin. So omeprazole induced the expression of CYP1A2 specifically, and rifampicin induced the expression of CYP3A4 specifically. As we can see here, the treatment of these two drugs do induce a very specific response of these two enzymes. Okay, let's look at the endothelial cell differentiation. So we stain the cells after sorting them for CD31, and we show that the vast majority of the cells are positive for CD31 and icadherin, as you can see here on the flow cytometry chart, and also on immune fluorescence staining. But the cells that we derive and also express von Willebrand factor, which is an endothelial anticoagulant factor. And once we plate the cells on matrigel and on a matrigel bed, the endothelial cells spontaneously start to make blood vessels, capillary vessels, as we can see here on image J. And also, the endothelial cells have this specific ability to uptake acetylated LDL, as we can see here on letter K. And for looking at what type of endothelial cells that we generate, we need to check the expression of arterial or venous marker. And we observed that CD31 expression is broadly being expressed in our cellular population, as we show it in flow cytometry chart, but we also see the expression of important arterial markers such as ephrin B2. And when we treated the cells with an inflammatory stimuli such as TNF-alpha, endothelial cells upregulated ICAM1, which is recapitulates the neutrophilic activation of the blood vessel. So that shows that our cells are indeed responsive to inflammatory stimuli. And finally, let's look at the mesenchymal differentiation. So we stain our cells at the end of the differentiation for mesenchymal markers such as CD90 and CD73, but we also check the expression of endothelial and hematopoietic markers such as CD31 and CD45. So we show that there's basically no contamination whatsoever. On the bottom image, we can see the expression of another stromal marker, such as CD105, and we differentiate the mesenchymal cells towards osteoblasts, adipoblast, and chondroblast, showing that the mesenchymal cells have the ability to differentiate towards these stromal phenotypes, as they should, because they are multipotent stem cells. So on the osteogenic differentiation, we stain the cells for alizarin red, as we can see here. For the chondrogenic differentiation, we stain the cells for Gaussian blue, which stains glycosaminoglycans. And for adipoblast differentiation, we stain the lipid droplets with oil red. So basically, we are showing here that the mesenchymal cells that we derived are indeed multipotent. And the expression of important osteogenic markers such as BGLAP, chondrogenic markers collagen 2A1, and adipoblast markers such as LPL are indeed expressed at the end of this differentiation process. So finally, just let me show you about the 3D hepatic differentiation process that we established in our laboratory. So basically, we applied the same recipe that we use for 2D hepatic differentiation, but starting off, instead of plated 2D plate iPS cells, but we start off as embryonic bodies. So the embryonic bodies are these 3D constructs that are derived when you take the iPS and start spinning them. And we do that by applying orbital shaking throughout the differentiation process. So basically, we use the same recipe, and we check the expression of important endoderm, hepatoblast, and hepatocyte markers throughout the differentiation process. When we observe the same pattern as well as we observed in the 2D differentiation process, as we can see also on immunofluorescent staining here on the right. So at the very first day, the embryonic bodies express pluripotent markers such as OCT4 and SCCA4, definitive endoderm markers at day 3 such as CXCR4, FOXA2, and at the end of day 9 of the hepatoblast differentiation process, the vast majority of the spheroids that we derived express alpha-fetoprotein and MEK adherent. And at the very end of the 3D differentiation process, the hepatic spheroid is stained positive for CYP3A4, albumin, alpha-1 antitrypsin, cytokeratin 18, and so on. So, in order to put these spheroids in the hydrogel and perform the extrusion and layer deposition using the 3D bioprinter, we need to make sure that the spheroids that we derived could fit in the needle, so they won't get stuck when they get extruded throughout the needle. So the mean diameter of the spheroids that we have was approximately around 200 microns. So it's very easy, no big problem to incorporate in a hydrogel and perform extrusion-based bioprinting. So these spheroids are actually hollow structure, and they have this internal basal lamina where the epithelial cells are deposited onto. So this is a very interesting feature of epithelial spheroid differentiation. So indeed, the cells are in close contact to each other. So, okay, we then go to the bioprinting process. We use a very interesting bioprinting process using this Incredible Plus bioprinter. We incorporate the cells, divided them into two groups, a single cell printed group and a spheroid printed group. And then we bioprinted them using extrusion-based bioprinting, as we can see here in the bottom left. And these spheroids are not damaged throughout this extrusion process, as we can see here on the bottom right image, after bioprinting the spheroid as well. So let's see how these two experimental groups perform over long periods of time in culture. So first we need to check the viability of the construct, so if the cells do survive over time. And then when we compare a single cell group to a spheroid group, we observed that the viability of the single cell group decreased over time significantly, as we can also see on the LDH expression activity, which is very high at the very first day of the single cell group and starts to increase back at the end of our 18-day period of culture. Important liver functional markers such as the albumin, which is being secreted in the media, also alpha-1 antitrypsin and other markers are significantly more expressed and secreted in the spheroid group as we compare to the single cell group. And finally, when we look at the expression of important hepatic markers, such as nuclear receptor 1-2, CYP1A2, CYP1A1, CYP3A4, and GSTA1, all of these markers are significantly more expressed in the spheroid group at the end of our protocol. So that shows us that in terms of hepatic function, the spheroid group are significantly superior as we tested and as compared to the single cell group. Okay, but what is going on with the single cell group? To answer that question, we performed a metabolomic analysis. So we took the supernatant of the culture and analyzed every metabolite on the media. And we observed on this principal component analysis a very distinguished pattern between single cell group and spheroid group. And when we look at the GOs of this analysis, we observe that urea and amino acids metabolism are the main differently observed metabolites in the media. So basically, the spheroid group has a lot of amino acids that are being generated in the culture. So why are these amino acids being generated in the culture? So to answer that question, we perform a proteomic analysis. We look at the differently expressed proteins on these two groups, and we observe that the most enriched pathways are associated with protein metabolism. So basically, what we show here is that on the single cell group, the cells are starting to break down proteins in order to generate energy. So this is not a very good feature because, as we all know, the first option to generate energy from is sugar. And the second one is lipids. And finally, if you do not have any more options, you start to break down proteins in order to generate energy. So that shows us that the single cells printed within these hydrogels are in a severe stress environment, and they're starting to die. And we check the expression of important epithelial mesenchymal markers and to see if there is any EMT, epithelial mesenchymal transition going on. And we observe that the vimentin expression is significantly being overexpressed in the single cell group. And vimentin is a mesenchymal marker. And also we test the expression of important epithelial cadherins, such as CDH1 and CDH2. And we observe that in on spheroid group, we can see significant increased expression of these two epithelial markers. And we test the expression of SNAIL2. So SNAIL2 is a transcription factor involved in the epithelial mesenchymal transition. And we see that the single cell group do express more of this gene. So combining all this evidence, we show that on the single cell group, the cells are starting to die. They are breaking down proteins, and they are indeed transforming towards a more mesenchymal, non-functional phenotype. So in summary, we establish a multi-differentiation process from a same patient using simple blood sample collection. We combine that with modern 3D bioprinting process, and then we generate a more mature, stable liver tissue that could one day be used as a graft for organ transplantation replacement. This organ is indeed autologous to the original patient, And this whole process today take us around 90 days, from the blood collection to the final maturation of the bioprinting liver. So in terms of future perspective in the bioprinting field, I consider that the development and the testing of BioInk formulae that uses liver-specific extracellular matrix proteins are very, very important. So we need to create the proper liver environment for the cells to perform as they should. And also we need to increase cellularity, which is the density of the cells that we use in the bioprinting strategies. Such as we can also apply new methodologies that have been developed over the last few years that do not use actually hydrogels. So they indeed generate a very high cellularity tissue. These technologies have been developed by several groups, and one very interesting recently published paper described this new methodology called sacrificial writing to functional tissue. It's a very interesting methodology that I believe that could be the next strategy for deriving liver tissues. But we also need to increase liver cellular phenotype diversity. The liver is not only composed by hepatocytes, endothelial cells, and stromal cells. The liver also has cholangiocytes, Kupffer cells, and so on, many other cellular phenotypes. And as we increase this cellular diversity, we should increase liver functionality. And finally, we need to increase structural complexity and size, because we need a very functional tissue, but also large tissue to perform the hepatic function that the patients with chronic liver disease needed. So at this point, I just would like to thank you very much for your attention. Of course, the work that I presented here was not solely performed by me. It's a teamwork. Especially, I would like to thank Professor Mayana Zatz, which is the PI of our laboratory, but also Professor Silvano Haya, Professor Peter Lelkas from Temple University, our longtime collaborator. But also, I would like to extend my knowledge to Dr. Luis Caires and Kaique Teles of our liver tissue engineering team. And also our colleagues from the Institute of Bioscience and the Medical School of the University of São Paulo. And also, I would like to thank our financial support for several institutions that made this work possible at the very beginning. So thank you very much for your time. And if you want to contact me directly, you can just scan this QR code and then contact me throughout all my social media links that you can see, that you'll find on this QR code scan. Thank you very much for your time and consideration.